So hello everyone and welcome to our Signature Square program from 1 Million Solutions in Health. We're really excited to have you here today. We're going to be hearing a fabulous talk from the NIH in regards to a vaccine against polyomavirus-induced brain disease. So we're really excited to hear from Dr. Chris Buck, and I'm just going to give you a little bit of an introduction to start off with, and then we'll jump into his presentation. Just from a logistics perspective, if you could put any questions you have for me, or not for me, to me, if you could choose my name where it says send to, so choose Dawn Van Dam. I'm the moderator for today, and I'm the president of 1 Million Solutions in Health. If you could put into the chat box any questions to me, and I will then read them out to Dr. Buck after he finishes his presentation. And at the end of the webinar, there will also be a brief survey that we'd like you to also fill out, so that's just like a heads up for you so you know that that's coming. And I think we've actually just stuck it in the chat box now so that you can grab it. So we're going to go through a brief introductions, and then Dr. Buck will do his presentation. We then would like you to do the Signature Square evaluation, which is in that survey, and we'll have some closing remarks at the end. So at 1 Million Solutions in Health, our goal is to improve healthcare one solution at a time, and we really are so thrilled to have this partnership with the NIH because this gives all of us an opportunity to see some leading edge science and help bring it to market and help patients get the things that they so dearly need in many different scientific areas. We fundamentally help connect people, we help learn and share, we bring innovation forward, and then through our technology evaluation consortium with the pharmaceutical industry, we help people collaborate to get to the end result quicker and make sure that we have a viable product to bring to the market. Our vision is to accelerate the discovery, development, and delivery of innovative cures, treatments, and preventative measures for patients around the world. And we basically have what we call a Hidden Gems program. We're helping you as the people who are in the entrepreneurial world get the edge in our marketplace for science by bringing you this science more quickly, like the science we're bringing you today from the NIH. We have all sorts of ways of bringing you science. I won't go into those details, but you can see in that second box is we're bringing you science through collaborations and licensing opportunities through the NIH. In our technology evaluation consortium, it's really a collaborative partnership between the pharmaceutical and biotech industry and other stakeholders. And really, here again, we're streamlining new technology considerations for this particular industry as well, too, and helping them do not just their evaluations, but their validations of this new technology to bring it forward. I'm going to skip that for a second. So these are just some of the people who have been part of our consortium over the years, a stellar group of people, and we've been so pleased to have them be part of our organization. And so really, fundamentally, at 1 Million Solutions in Health, we help find new scientific solutions. We gather input to optimize the technology, and we share the findings, and that's what Dr. Buck is going to be doing with you today, is sharing the findings that he has in order to help accelerate and move it forward to a licensing or collaboration opportunity. So just a little bit about the Signature Square. I'm going to jump, I think, to our picture slide. Is really, we help understand best where the technology is situated. Is it a blockbuster or is it novel or an upstart? And we really help kind of bring things forward to the marketplace as quickly as possible. For NIH scientists, and why this makes sense, is we have been looking to bring more collaborations together, more licensing agreements. So the #1 goal is to have a licensing agreement come out of this presentation and have someone say, yes, I want to bring this to market, because this is fabulous science. Sometimes the NIH is also looking for creatives or research and development agreements. So that's also sometimes the purpose of one of these talks, but today it's really about licensing agreements. And sometimes we're also looking for feedback into the NIH roadmap. But today we'd like to get this to be a licensing agreement. So with no further ado, what I'm going to do is I'm going to turn it over to Dr. Buck to give you today's presentation. As I mentioned, put any questions you have in the chat box, and we'll be sure to answer them at the end. And I'm going to pass the ball over to Dr. Buck. And what I'll do is, before he goes, I will just do a very brief overview of his background as he gets ready. So give me 1 second, and I'll do that. So the presentation today, just to give you an overview of that first, Polyomaviruses are a family of tumor viruses that cause brain and kidney damage in immunosuppressed individuals. The polyomavirus protein spontaneously self-assembles into virus-like particles, or VLPs, that closely resemble the native virions. VLPs have proven to be exceptionally potent vaccine immunogens in humans. The NCI has recently been allowed patents, so these are like hot off the press, covering the use of VLPs as vaccines, targeting two human polyomaviruses known as JCV and BKV. In this webinar, NCI Senior Investigator Chris Buck describes the application of the polyomavirus VLP vaccine to the prevention and treatment of JVC-induced brain disease. Patients on a variety of immunosuppressive therapies, including the successive multiple sclerosis drug, sorry, I'm having trouble with that this morning, natalizumab, constitute a well-defined market for the polyomavirus vaccine. Since VLPs are generally safe and patients have no other option for preventing this devastating disease, the vaccine is likely to be eligible for FDA fast-track licensing. So that's really, really exciting that perhaps someone who would license this would be able to get it through the FDA in a fast-track way. So I'm just going to do a really quick intro on Dr. Buck. Dr. Buck received a bachelor's degree in molecular, cellular, and developmental biology from the University of Colorado at Boulder. He then returned to his home state, Maryland, to earn a PhD from the Johns Hopkins School of Medicine. Dr. Buck's graduate research in Robert Feliciano's lab focused on the translation and immunogenicity of the HIV capsid protein GAG. For his graduate work, Dr. Buck received the Alicia Showalter Reynolds Award. In 2001, Dr. Buck began postdoctoral training in the lab of cellular oncology, where he developed systems for producing human papillomavirus-based gene transfer vectors, or pseudoviruses. His work using HPV vectors ranged from basic studies of HPV virion structure and morphogenesis to translational research identifying candidate topical microbicides for blocking HPV transmission. For his work in these areas, Dr. Buck and his world-renowned mentors, Dr. John Schiller and Doug Lowy, shared the 2006 Norman P. Salzman Award. In 2007, Dr. Buck joined the faculty of the NCI Center for Cancer Research as an investigator. In 2013, he shared a second Salzman Award, which is absolutely fabulous, in recognition of his mentoring of Dr. Rachel Showalter. Dr. Buck was awarded tenure in 2014. So you've got, I think, one of the most world-renowned and most specialized people in this area talking to you today. And I know, because I've seen the presentation already, that it's just fabulous information and research, and you're going to be really very impressed. So with no further ado, I will turn it over to Dr. Buck.
Thank you, Dawn. I'm literally blushing a little bit from this flattering introduction. So I hope I will live up to the invitation to wow the audience. So as Dawn was saying, I trained with two of the guys that invented the current HPV vaccines. We studied papillomaviruses in that work. And then when I branched off into my own lab, I reoriented toward a different group of viruses called polyomaviruses. And as you can see from this these images in slide 2, these are actually quite similar viruses. They look exactly the same from the outside, and they used to be categorized in the same viral family. Their organization is a little bit different, and there are aspects of their biology that are different, but many commonalities exist. So the papillomavirus major capsid protein, which is called L1, can self-assemble into a particle that looks exactly like the normal native virion. And so these particles are the basis of the NCI's HPV vaccine technologies that have been so successful. A thing that we've learned from the HPV vaccine is that these VLPs are incredibly immunogenic. So the current trials for the HPV vaccines are showing that a single dose of these particles can elicit neutralizing antibodies that are 1,000 times more potent than they need to be to protect women and men at this point. And the even more encouraging thing is that the durability of the responses is essentially permanent. The titers are flat. These people will have neutralizing antibodies for the rest of their lives. So these VLP vaccines are a technology that really needs more attention, and some of the technology is being reoriented towards other diseases and other pathogens where you need these high titer neutralizing antibodies. But one thing that is really low-hanging fruit is the cousins of papillomaviruses, these polyomaviruses that my lab now studies. So BK polyomavirus was discovered in 1971. It's so old that BK actually is patient initials, which we don't do anymore. So this was the first human polyomavirus. Its major capsid protein, which is called VP1, just like the L1 of papilloma, assembles into particles that look just like the outside of the normal native virus, and they are similarly highly immunogenic, just like the HPV VLP vaccines. In fact, if anything, they're even more potently immunogenic than the HPV particles. Okay, so polyoma is Greek for many tumors. The original mouse polyomavirus can cause many different types of cancer in experimentally immunized animals. There are now 14 known human polyomavirus types, and we believe that basically for all 14 of them, everybody gets infected, and the infection is lifelong. It's not associated with symptoms that we recognize, at least. It could be causing diseases that we haven't figured out yet. But there are some diseases that we know these viruses cause. So in immunosuppressed people, particularly kidney transplant patients, but actually other types of transplant patients as well, BK and to a lesser extent its cousin, close cousin, JC polyomavirus, are both a cause of kidney disease in the engrafted transplant recipient. So I'm not going to talk about the kidney disease today. That's for a later webinar. I'll touch on it a little towards the end of the talk. But today we're going to talk about a different disease that's mostly caused by JC, although sometimes also caused by BK, and that's a highly lethal brain disease that also strikes various types of immunosuppressed patients. So PML is the brain disease caused mostly by JC polyomavirus, and it was a hallmark feature of AIDS. It was a classic AIDS-defining condition, and so it is a condition that occurs when the immune system stops surveying the brain for viral infection, and the JC replicates out of control, and this causes many sites of damage to the white matter of the brain. Other more recent immunosuppressive therapies have also been turning up with PML as a side effect. And so the prime example is a drug called natalizumab. The trade name is Tysabri. And this is a monoclonal therapeutic that basically stops T cells from getting into the brain. And this is good news if you have multiple sclerosis, because T cells are causing damage in the brain. But as soon as the T cells stop surveying the brain for viral replication, you become at risk for unchecked replication of JC, and this can lead to PML. And this is a sort of a 1 to 2% risk of people on natalizumab, and the risk, it's increasingly looking like it may be 1 to 2% per year that you're on the therapy, particularly for certain subgroups of patients. So natalizumab right now is probably the best currently available therapy for multiple sclerosis, and there are people on it. It was actually briefly withdrawn from market, and the FDA had contentious hearings about putting it back on the market. Multiple sclerosis is a very bad problem, and so people testified at these meetings, yes, I understand that there's a risk of PML, but this is the only drug that gets me out of the wheelchair, and I'll take the risk. So it's on the market, and people are rolling the dice, and it's a very grim and difficult situation. It's a very profitable drug. It costs about $70,000 a year to be on it, and the gross sales are about $2 billion a year. So about 2% of these patients develop PML between the first and second year of therapy, particularly in certain subgroups. And the use and broader application of natalizumab has been severely limited due to these side effects. So another target of natalizumab is things like ulcerative colitis, but there you would not risk brain disease to treat a condition that you can live with. PML is also a side effect of other drugs. In some cases, the PML cases just show up because these are people who are immunosuppressed for other reasons. But natalizumab is clearly causally associated with the side effect. It's not just an indication that you have an immunosuppressed population. So this has led to black box warnings for some drugs, of course, natalizumab, obviously, but I'm having trouble advancing the slide. That's a puzzle. Another major problem is with the wonder drug rituximab or Rituxan. This is an anti-B-cell drug that's good for treating B-cell lymphomas and also rheumatoid arthritis, and there have been a couple thousand PML cases reported to the FDA of people on this drug. So this is a big problem for this second drug. Some other drugs have PML warnings in the black box, and one drug called Reptiva that was against psoriasis has had its license revoked, and it is now permanently off market because of PML side effects. So even for drugs that don't have known PML cases, like this drug Entyvio, there is kind of a shadow over the community. So Entyvio is a cousin of natalizumab. It just stops the T cells from getting into the gastrointestinal tract and is not known to stop T cells from getting into the brain. So you could imagine that Entyvio might not ever cause PML because its mechanism of action is different. But this didn't stop the FDA from demanding that the TV commercial have a mention that this is in a class of drugs that has PML as a known side effect. So in the TV ad where this nice lady is going off to yoga practice, the announcer says, PML, a rare serious brain infection may be possible. So I have an old college roommate who has ulcerative colitis, and he told me he is not considering asking his doctor to try Entyvio because rare serious brain infections don't sound like a good thing. Okay, so the solution to this problem that has emerged is that it should be possible to vaccinate people against JC polyomavirus, the virus that causes PML, and this should protect them against the disease through neutralizing antibodies. So our lab has pioneered the basic science underlying how this vaccine would work. It's been found that JC polyomavirus found in the brains of PML patients carries a distinctive set of mutations, and these mutations are in a very suspicious place. They're right near where the cell surface receptor glycan docks on the surface of the virus. So this is how the virus attaches to cells, for example. And so these mutations are right in the place where you would expect maybe neutralizing antibodies would bind. And indeed, that's the case. These mutations allow the virus to escape from antibodies that would neutralize the wild-type virus. So this is a case study illustrating that point. In this particular PML patient, we had samples from before they developed PML. They went on to die of the disease ultimately. And you can see that at the beginning, they failed to neutralize the mutant virus that was found in their brain, the 269F mutation specifically, even though they neutralized the wild-type virus. So this mutation allowed the virus to escape from this one patient's neutralizing antibody response. And this patient died being unable to neutralize the virus that was killing them. Things went differently for a patient who survived. So patient 5040 on the right was neutralizing both wild-type and mutant fairly poorly at the time they were diagnosed. They eventually started neutralizing the wild-type, but not the mutant. But they survived PML, and they went on to eventually develop neutralizing antibodies that cross-neutralized all of the viruses. So it's not that the mutant is intrinsically resistant to neutralization. It's just resistant to the neutralizing antibodies that were present in this one patient at the time that they were developing the disease. And so this shows that people can mount good, broadly protective responses that would protect them, but these patients who are developing PML just don't have the right antibodies at the time that they develop PML. So another another case study involves a compassionate use license that was granted in Europe, and there was a team that already had JC polyomavirus VLPs made, and they got authorization to administer these VLPs to some patients who were really in desperate shape from PML. So in this case, it was a 75-year-old woman. She had PML due to CD4 T cell deficiency of unknown cause. It was not AIDS. It was just that she had poor CD4 T cell function, and this led to PML. And soon after she was diagnosed, she was given these JC VLPs on an experimental basis. So you can see we don't have samples from before she developed PML, but you can see that she started off not neutralizing her mutant virus very well. This is her cognate mutant, even though she neutralized the wild type quite well. You can see that after vaccination, her titers spiked up for both of the viruses, and don't be fooled by this log scale here. Her titer went to 25 million. We think this is the record for the highest antiviral neutralizing titer ever observed in a human subject. It almost defies the laws of mass action, and this again is a 75-year-old woman who is CD4 T cell deficient. So this again just reinforces the point that these VLPs are incredibly potent vaccine immunogens, and it shows that they can elicit the cross-neutralizing antibodies that would have protected this woman against the PML mutant that was replicating in her brain. So this patient, by the time she was vaccinated, she was already in a coma, and brain damage is irreversible. But the vaccination did correlate with an arrest of the very aggressive progression of the disease. So she's still alive and in a coma, and so this is not necessarily an appealing therapy. This is something you would want to do before the brain damage occurs in the application that we're proposing. So PML is a rare disease. There are currently no very effective treatments aside from just immune reconstitution. Immune reconstitution comes with a high risk of immune reconstitution inflammatory syndrome, which is itself sometimes lethal. So the prognosis is quite bleak for this disease. We know that VLP vaccines don't generally cause harm in human subjects. We know this from the HPV vaccine and also from this small set of 6 subjects who got the compassionate use vaccination. And so this presents pretty much a perfect fit for the FDA's new guidelines on fast-track regulatory licensing. And so what this would allow for would be to basically just do a safety trial and then go straight to market. You could conceivably sell the vaccine to anybody who believes that they might be at risk of PML, obviously natalizumab patients, and maybe some other patients, and basically this would be on market, and you would look retrospectively for efficacy. So the path to market is basically first develop a GMP pipeline for the VLPs. Our lab has actually already done this. We have some GMP-compatible VLP stocks that, as I'll tell you in a minute, we're just about to put into monkeys to qualify them. There will probably need to be a human safety trial, but this actually could just be healthy subjects who volunteer for JC VLPs. There's no reason to believe they would cause any harm. And after the safety had been established, one would go straight to the PML prevention market. The obvious targets would be people who are on natalizumab. There's a paper showing that people who are on natalizumab are perfectly able to respond to vaccination. There's nothing wrong with their peripheral T cells, just T cells trafficking into the brain. And patients who are considering rituximab could, of course, also be a hot target for JC VLP vaccine. Conceivably, there would be people like my old college roommate who might get the JC VLP vaccine and then try Entyvio. And so if this type of market evolved, it could really be a colossal market for the vaccine. Anybody who has HIV infection should, of course, be on combination antiretroviral therapy, but therapy does sometimes fail, and you certainly would not want PML to be the first sign that you're failing therapy. So if I had HIV, I would be clamoring for a JC VLP vaccine, just as an insurance policy against PML developing at some point in the future. Okay, so the efficacy would be established in this scenario just by looking backwards and asking whether people who receive the vaccine get less PML than the historically established rates. And these are very well established numbers for natalizumab. It would not be difficult to look backwards and say, oh, these vaccines have less than we have been seeing in the past, because what we see with people on natalizumab is the subject of at least a dozen different really well-conducted studies. Okay, so I haven't told you anything about the other disease these two viruses cause, which is nephropathy, particularly in kidney transplant patients. In that application, which I'm going to talk about in September, there would need to be an efficacy trial up front. I don't believe that one would be eligible for fast track, but the trial is very simple and would probably only involve a couple dozen subjects, and it would allow the exact same vaccine that had been allowed in the exact same safety trial to be retargeted for this second application. Okay, so that market, again, I'm going to enthusiastically tell everybody about on a September 12th webinar, but the landscape is that this nephropathy is a big problem in kidney transplant patients. About 20% of patients become BK viremic, and what I neglected to say is that you must test kidney transplant patients for BK in their blood. This is standard of care, and that's an expensive test that requires physician visits. There are about 30,000 people signing up for kidney transplant every year in the U.S., and that's when you would vaccinate people, as soon as they sign up for an organ. So this is also a fairly large market. And one argument for having a combined vaccine that has both JC and BK comes from our studies with these GMP-compatible VLPs that we've developed. This is neutralization results from mice, but you can see that mice on the far right, mice that received JC VLPs neutralized JC very well. That's the bars on the far right. After priming and boosting, they were neutralizing JC with titers of 1 million. But those same mice, interestingly, cross-neutralized every BK that we tested. So these are, again, very close cousins, these two viruses. And there's actually a side benefit of including both BK and JC in the vaccine, because you just get higher cross-neutralizing titers against both of the viruses that you're targeting. So we've immunized monkeys as of last month. It'll be a couple more months before we have the neutralization results for the monkeys, but we're optimistic that monkeys will be like mice and people, and these VLPs will be extremely immunogenic in them as well. Okay, and I am now going to pass the ball over to my colleague, Joe Conrad, who has almost infinitely greater expertise on this licensing stuff on this slide than I do. I think I passed the baton.
Yes, you did. Thank you, Chris. I appreciate it. So hi, everybody. My name is Joseph Conrad, and I am a Technology Transfer Manager with the National Cancer Institute's Technology Transfer Center. I'm stepping in just to give everyone a quick summary of what we have available from a patent perspective. So to reiterate Chris's earlier comments, the BK and the JC vaccine that's presented here can protect against both kidney disease, which he just briefly touched on, and which he'll touch on in more detail next week, in transplant patients, but also in the current PML presentation as well. So for those interested in potentially licensing this technology, we just were awarded or allowed claims in both the U.S. and Japan. The U.S. claim coverage covers a method of eliciting an immune response with the trivalent BK and JC vaccine. And you can see the trivalent nature in the parentheses there. And that's referenced in our reference number E168-2011. And that number can be searched on our website for a brief abstract of what the technology is, or you can contact Kevin Chang, I'm sorry, at the end of this presentation as well. So if you want more information, his contact information is on the last slide. And in Japan, we have vaccine compositions to that trivalent vaccine as well. Same reference number. So if we go to the next slide, these are the claims that are pending, currently pending in both the U.S. and in a current PCT application that's being evaluated. So for the pending U.S. claims, we have vaccine compositions directed to a trivalent vaccine, again, same reference number. And in the PCT, we have monoclonal and polyclonal, or polyvalent rather, polyvalent and monovalent JC vaccine compositions and methods of using them to elicit an immune response in patients. And so that reference is our E549 2013. So again, you can search those numbers, those reference numbers in our website and get a brief abstract of what the technology is, and for more detailed information about the technology, you can contact Kevin Chang, who will set up a meeting, conversation with Dr. Buck about this technology in more detail if anyone would like to pursue it further. So with that, I will pass the ball back over to Dawn, our host, and she will continue with the webinar.
Great. Thanks so much, Joe. Really appreciate that. And thank you very much, Dr. Buck. I thought that was fabulous information and very, very clear and very straightforward as to the opportunity. So maybe just, I'm going to invite the audience to send in any questions to the chat box. And maybe just I'll ask questions first to Joe. Joe, I'm sure lots of people do understand the patent law, but maybe some people don't. Can you just explain, right now, as I understand it, the patent has not been issued, but what's the difference between what has occurred and a patent being issued?
So in the patent office, in the patent process of examining a patent application, once a patent application is filed, it goes to an examination process. If the patent application is going to be issued into a patent, the first stage is to allow the claims that the application has presented to the examiner for evaluation. So we've gotten to the stage where the claims have been reviewed by the patent examiner, and the patent examiner has said, yes, these claims are allowable, meaning they will become a patent in the next stage. So once the claims have been allowed by the patent examiner, that goes back to the patent attorneys who represent the client, in this case us, and there are certain hoops that the clients have to jump through to move to the next step. Will move the patent to its issued status. So once the patent is allowed, once the claims are allowed, basically, and the examiners, not the examiners, but the attorneys have completed the next paperwork-related steps that are necessary to allow the patent office to issue a patent. So we are one step away, basically, from a issued patent that has a patent number, and there's nothing stopping that from moving forward on our end. So this is essentially a pro forma type of process that at some point after, well, at some point beyond this point, they will issue a patent.
And so it's basically a done deal, is what you're saying, that a patent will be issued?
Yes.
And so we're getting like an early warning of that information. Is that correct?
Right. It's a heads up that, hey, this is an allowed patent. We will move this to issuance, and we will issue the patent, but we need to dot the I's and cross the T's from the point we allow it to the point we issue it. Okay.
And so any thought that you can give on, like, what's a typical time frame that would happen?
That I cannot speculate on, no. It's going to come. And Kevin, who is also a technology transfer manager in the office, and I believe he's on the line, may be able to give that information. But the patent process.
So, Joe, let me interject. This is Kevin.
Hey, Kevin. There you go.
Hey, sorry. Yeah, actually, the issuance is going to come fairly soon, and probably within, the next month or so. We've already paid the issue fees, and so, we're just waiting on the patent office to send us the paperwork to show that it's actually issued as a patent.
There you go.
Awesome.
I couldn't do
Fabulous.
it, but Kevin could.
Yeah. And what do we expect the length of the patent to be?
So
like how many
there's approximately 15 years of patent term remaining.
Fabulous. Great. Excellent. And so a fast track through the FDA with 15 years and the information, the patent should be issued in a month. I think that sounds like it's pretty hot.
Absolutely.
Okay, great. So some people have submitted a few questions here, so let me jump over to those questions. So this is for you, Dr. Buck. Do polyomaviruses really cause cancer in humans?
So yes, this is a question that is at the front of my lab's mind all day, every day at this point. So the case that is the farthest along at this point is BK polyomavirus in bladder cancer. Bladder cancer is the 6th most common cancer in humans, or in the U.S. at this time, and so that is a big and important problem. The problem is that when you look at bladder cancers that are, metastatic disease that is killing people, in the general population, the virus is rarely in the cancer. So we have a giant problem in the field, which is that what we call the hit and run hypothesis. And in this idea, the virus is there as the tumor is developing, and it's causing the cell to grow and survive all of the oncogenic damage that it has to survive to become a cancer. But then later in the process of cancer development, the virus is just lost. It becomes like training wheels that is no longer necessary for the tumor after it's accumulated enough hits. And so this hit and run problem is a giant problem. People's careers have been sacrificed on the altar of this intractable question. But what's been happening lately is that we've been finding that kidney transplant patients who we know have a problem with BK polyomavirus much more often have BK integrated in the finished bladder cancer. And so we know that sometimes, at least in those patients, BK is causing cancer in a way that you can detect it in the tumor. The epidemiologists have been helping us out a lot too. So it turns out that if you have a kidney transplant, you're at about 4-fold increased risk of developing bladder cancer and kidney cancer as well. But then some recent studies that have just been coming out have looked at the question of whether a kidney transplant patient who has BK viremia is at even greater risk, and the answer is yes. If you had a kidney transplant and then you got BK viremia, you are at another 10-fold increased risk of getting bladder cancer relative to kidney transplant patients who didn't get BK viremia. So despite this hit and run problem, we have already kind of arrived at territory where the evidence of BK causing at least a small fraction of bladder cancer is as good as evidence for other cancer virus connections like Merkel cell carcinoma or even HPV in cervical cancer. And so that was a very long answer that I think is yes, I think that these polyomaviruses are probably causing cancer in people. And the last thing that I'll add is that there is one human polyomavirus that I just alluded to. This is human polyomavirus 5, also known as Merkel cell carcinoma. And that virus was discovered as an integrated virus in a kind of skin cancer called Merkel cell carcinoma. And so in that case, human polyomavirus 5 in the skin disease Merkel cell carcinoma, the causal connection is widely accepted. Everybody believes that that virus is causing a form of skin cancer. And so the job going forward is to now find out what other cancers these viruses might be causing in this very difficult to address kind of like hit and run scenario.
Okay, and since the people on the webinar today who are listening are interested in the PML side effect for patients who have multiple sclerosis or ulcerative colitis or other related diseases, does the cancer piece matter to them at all, or is it just interesting scientific information that says this virus is even more important than we thought?
So, today, the only cancer angle in today's talk was that rituximab is a cancer therapeutic, and there are several other cancer therapeutics that you'd want to vaccinate people against PML before they go on this cancer therapy. In the longer view, this will be a vaccine that will have both BK and JC, and so you know, actually some JC has shown up in these bladder cancers that I was talking about in the kidney transplant patients. So the reason that anybody developing this vaccine should be interested in the question of cancer is because this could be the vaccine against bladder cancer. I mean, basically that's the goal that my lab is now working on, is to establish the connection between BK and JC and bladder cancer, and then everybody should get this vaccine because bladder cancer is the 6th most common cancer in the U.S.
Okay, so from a business perspective, if I were someone who was interested in licensing this vaccine, I could either license it just right now for the more narrow indication of PML, but perhaps I'd be building a relationship to work with you in the future to utilize this vaccine in other areas, but that's not a requirement that I have an interest in that in the future. It's just something that might be interesting to know, correct?
Yeah, so the connection between these viruses and cancer is not well enough worked out for us to even try. The place actually, here's where they're connected. The place that this information will come out is that the kidney transplant patients that I'll talk about next time will get this vaccine, and we know that they're catching bladder cancer in the course of just a couple of years. So this hit-and-run problem is completely intractable at a basic science level, but if we vaccinate some people against BK and JC and they get less bladder cancer than we would have expected based on historical baselines, that will be the conclusive proof that these viruses are causing cancer. So in other words, anybody who develops these vaccines is going to have to agree to just look at people and find out whether they get less cancer, because that's the best experiment that I can think of to establish the causal connection.
Okay, so just again from a business perspective, if anyone who's on the line is listening and saying, okay, I'm only interested in PML as a side effect for multiple sclerosis or ulcerative colitis patients, that's okay. The only commitment I might have would be just to, as you say, look at cancer incidence going forward, but I could still be narrowly focused in my licensing of the technology and my go-to-market strategy.
Yeah, and you're stirring up a side issue that I probably should get into, and that is that I don't think that it makes sense to have a JC only or BK only VLP vaccine. I really think this needs to be a 3-valent vaccine. And I really think that it's equally applicable both to the kidney transplant population and the PML population. So what I, as a scientist, I mean, I can't speak for the NCI, but what I, as a basic scientist, would like to see is that whoever licenses this technology is planning to apply it to both the, I predict, highly profitable PML market, and I predict the highly profitable kidney transplant market. I think it's one vaccine that has 2 applications that really both need to be pursued. So, you know, it's not me making the licensing decisions, but if I had a vote, I would say whoever licenses this really needs to look at both of these webinars. They need to think about both of the applications of the vaccine.
Okay. And maybe I'll just let Joe comment, but I think according to what Joe may have told me in the past, I think, you probably aren't part of the decision-making process.
Correct.
And
correct.
Also, not to scare off anybody, I think that the NIH does decide licenses separately. You know, it could be a joint package or it could be a separate package. So I don't want to scare anybody off who might be just interested in the PML. Is that correct,
Well,
Joe?
Dawn, I'm going to defer to Kevin on that because he's actually the person in agreement. Okay. So he's the better person to answer that question.
Right. So, Dawn, I mean, we certainly would, you know, any person or companies that are interested in licensing this technology would fill out a license application and submit it to me, and then we would take a look at the license application and see, you know, what they plan to do with the technologies, whether they plan to just develop the BKV application or the JCV application or both. But we would certainly take a look at all the information that's provided by the applicant in the license application. So, you know, we can make a determination after we see what is provided by the prospective licensee.
Great. And I think the most important thing is let's get a licensing agreement and get the product to market, right?
Right, absolutely, yep.
Yeah.
And,
okay,
and you know, it should also be pointed out that, you know, we're also open to collaborations, so it could be potentially a collaboration / licensing, that for both, for this technology.
Okay, awesome. That's great to know. Thank you.
I'll toss in that one thing I do have power over is what my lab does, and this is our top priority. So whoever licenses this vaccine gets free support from us to the extent that I can provide it. So, we would happily do things like neutralization assays, which we've pioneered, and help with the development of the GMP pipeline. I mean, the structure of these virions is what keeps me awake at night, and so we can throw resources in that direction using my lab's kind of basic core mission.
Yeah, and I think that's really a very important point about the GMP process, because that can often be a huge stumbling block for people, either because, it could be technically challenging or because maybe it's not exactly in their previous history and their bailiwick, so to speak. So I think that's actually a huge offer that you're making right there.
In a way, yes, the GMP pipeline can be a big stumbling block, but it is done. I think we have already solved that problem, and we're just looking for somebody to implement it at this point.
Yeah, that's monumental. That's great. Thank you for clarifying that further, because I don't think people would have known that necessarily. Okay, so a few more questions for you, Dr. Buck. You mentioned that PML is an AIDS-defining condition. Would HIV-positive individuals be candidates for this vaccine?
Yeah. So as I say, if I try to project myself into being HIV positive, I would be clamoring for this vaccine because PML is just one of the worst things that you can imagine, and so yes, certainly this is a potential market. I think if you establish efficacy against PML, I really think that PML is a known thing that people with HIV have to worry about, and so that really could be a part of the market in the long run, I think. I mean, how about this? We have pursued a project looking at whether JC polyomavirus is found in cases of elderly dementia. So there's some older literature that just found JC in just random people who died in car accidents. So you just look in the brains, and 20% of people over 60, you can find some amount of JC, and not just by PCR, but these papers had Southern and they had staining, and so we've all got this virus in our brains, and I don't feel good about that. Like, I feel like if I should just drift into immune compromise in some occult way, I don't want PML to be the indication that that's what's happening to me. So I could recommend, there's a Washington Post series on medical mysteries, and last year they had one on a woman who was about 63 years old, had no known risk factors, and they diagnosed her over the course of 9 months with having a series of small strokes. And finally, somebody thought to check her immune function, and it turns out she was idiopathic CD4 deficient. And then they put her in the MRI, and then they found out that what she had was not a series of small strokes, but it was PML. And so PML is a rare thing, but it does happen to people, and that's just the full-blown PML. Whether there are people wandering around with a little bit of PML, I wouldn't care to guess. So I think when this vaccine comes out, I might take it because I just don't want any chance of JC having wild parties in my brain. So, I mean, that gets us far beyond. Certainly if I had HIV, I would definitely want this vaccine. Even if I don't have HIV, I might want this vaccine.
Right. Okay. So again, people on the line may know the answer to this question, but so is there a treatment for PML or is the course always death?
So the course sometimes is spontaneous recovery. The treatment is immune reconstitution. So if you present and you come to the clinic and you have AIDS, the first thing that you do is put you on antiretroviral therapy and allow your immune system to come back and bring the virus under control. As I was mentioning, that also comes with the possible side effect of immune reconstitution inflammatory syndrome, and this is basically the T cells come back and then they see, oh, there's all this virus in the brain, and then there's inflammation as a result, and I think something like half of people die of that. And so yeah, there's a treatment, but it's not good news. And then in the case of that woman in the Washington Post medical mysteries, you know, she had idiopathic CD4 deficiency, so immune reconstitution was difficult. All of this is worth mentioning because it makes the efficacy trials a little complicated. Like, I don't think that I would want to give somebody the JC-VLP vaccine if they were diagnosed with PML AIDS, because the vaccine could theoretically exacerbate the immune reconstitution inflammatory syndrome. But for these people with idiopathic CD4 deficiency where there's no way to reconstitute them, they might be appropriate candidates for the vaccine. So all 6 of the people who were tested in the compassionate use trial were idiopathic CD4 deficient, and none of them seemed to have exacerbation in the kind of immune reconstitution inflammatory syndrome type of way. So that was a long answer. There are no good treatments right now is the short answer.
Okay, great. And I know you are not at the FDA, but you're probably very knowledgeable about what the FDA would allow and how everything would go forward. So if on the one hand you might be wrong about the fast track and the FDA demands a phase 2 trial, what might that look like, do you think, so that it would satisfy the FDA?
So for PML, it's a somewhat complicated trial. You would have to focus on natalizumab patients where the risk of PML is high and well established. And so that trial would basically get a group of people who had already been on natalizumab for a year. So usually they develop PML between year 1 and year 2. And you would take a group of perhaps a few hundred or 1,000 PML patients, vaccinate them and at year 1, and then wait for a year and ask whether fewer than 2% of them develop PML during the study period. So 2% is low numbers. This makes it for a difficult trial, but it's not impossible. It's just a difficult trial, and it would be somewhat expensive to conduct this trial because you'd probably end up having to pay for their natalizumab therapy while they're on study. So if the FDA, which it would be very strange if they did, but if the FDA demanded an efficacy trial up front before marketing, it's doable. It's time-consuming and expensive.
Okay, terrific. Thank you. So I'll just see if anyone in the audience has any additional questions. In the meantime, maybe I'll just ask Kevin or Joe, do you have any sort of further closing remarks or any advice or next steps? And then I'll ask Dr. Buck to give some closing remarks as well. So Kevin, anything from you?
Sure. So as I said before, if there are any companies that are interested in this technology, whether in licensing and or collaboration, certainly give me a shout out either through a phone call or through my email, which is changke@mail.nih.gov.
Great. I just put it back up on the screen again. Thank you. And Joe, anything from you?
Nothing more than to thank the participants for their attendance and interest in this technology. The group that I work with within the tech transfer office is responsible for helping to move this technology out of the NIH into hands of the private sector who can manufacture it and commercialize it. So we greatly appreciate the attention and interest in the technology, and anything that they can do to help get this to the patient population that needs it would be greatly appreciated.
Great, thank you. Well, Dr. Buck, it looks like you've done a great job answering questions, so no more new questions have come in, but do you want to just give some closing remarks, and then we'll close it out from there?
Yeah. I mean, I will second the notions that Joe just put forward, which is that these are bad diseases that are very troubling. I really sometimes I spring awake in the middle of the night thinking about the people suffering, and I think it's an easy path in a way to help these patients. I think that the path is now very well blazed, and I also think that it's going to make money. That it is not my job to think about, but it seems like that this should be a no-brainer for people who want to work in this kind of area of vaccine development. So I hope that we can attract some partners, and I look forward to supporting their noble efforts to move this thing forward.
Super, that sounds great, fabulous. Well, I'll just remind everyone again that there's a short survey. If you could just click on the link that's in the chat box, that would be awesome. And we really appreciate everybody taking a good listen to this and looking at and seeing if you can be the organization or person to help us bring it to market. Again, thank you very much to Kevin Chang, to Joe Conrad, and to Dr. Chris Buck for their help in getting this information out. And we're excited to see where it goes from here. So thanks again, everyone, and thank you particularly, Dr. Buck.
Great. Thank you, everybody.
Have a great day.